Hello everyone, and welcome to our online lecture series for Bio 2. Today we're going to continue on with our discussion of all things meiosis and focus a large part of the lecture today on what happens when things go awry, when things don't work out like they should. So, we'll start with a brief recap of what the products of meiosis are. We have these two drawings from your textbook, where you can see the different chromosomal rearrangements to generate our 4 different gametes. Keep in mind that both of these illustrations do not show recombination events, but in some cases, you'll get all of dad's chromosomes on one side, mom's on the other, and they will segregate after having aligned independently with that principle of independent assortment and then segregation to different gametes, resulting in 4 genetically unique, after accounting for recombination, gametes. In other circumstances, with that independent assortment, you can see that mom 1 or dad 1 might line up next to mom 2, and mom 1 might line up next to dad 2, giving you, again, different gametes. We illustrated this on our own, with pink indicating mom's derived, dad indicated by blue, and recombination illustrating the fact that all 4 of our gametes were genetically unique from one another. And we talked about that process of recombination, where the chiasmata forms, roughly 1 per chromosome arm, in order for a crossover event to occur, a recombination event to occur, so that meiosis happens without issues. But that doesn't always occur so smoothly. In some cases, that piece that was supposed to flip over from mom to dad and dad to mom, well, sometimes it goes missing. This is a double-strand break, after all. The DNA is floating around. If the chiasmata and the associated proteins do not hold on, you may encounter something called a gene conversion event. It looks a lot like recombination, but can you spot the difference? Hopefully you saw that both of the top arms are now blue. In this case, the pink, the mom-derived material that was supposed to stitch over to dad's chromosome, has floated away. But rather than leaving that chromosomal piece off and missing for eternity, the loss was detected and we were able to copy back the dad's portion back onto dad's chromosome. And in this case, mom's material for that segment has been completely lost and dad's has now been duplicated. Therefore, we call this a gene conversion event. Not because blue invades and takes over mom's without letting go, but more than likely because the pink segment was actually lost. So it does not come about from your intuitive sense of blue just being copied twice. It comes out because pink was missing, and therefore we copied blue twice. This is one example in which some of the segregation or some of the recombination or some of the chromosomal alignments in meiosis can go wrong. But a gene conversion event will fix things. After all, you don't know what's mom-derived, what's dad-derived. You just have alleles that are there. And the chromosomes go on to form their gametes just fine. But there are cases where the entire process of meiosis will go wrong. And in order to discuss those, we have to introduce the term called non-disjunction. A mouthful, I know, but non-disjunction refers to the fact that what happens in either meiosis 2 or meiosis 1 where the chromosomes are supposed to split apart, well, we refer to that as disjunction. In non-disjunction, you're instead seeing that the chromosomes do not split, and they travel together. There are two different cases of this. You can have a non-disjunction event that occurs in meiosis 1 or a non-disjunction event that occurs in meiosis 2. And you'll notice that the after effects are very different. So in the case of meiosis 1, what you're seeing is that the two sets of chromosomes migrate to one gamete. That leaves one set with double the amount and one set with absolutely zero. This means that the eggs that are produced from the set that has zero will have zero chromosomes, at least at that particular chromosome. You don't have to get non-disjunction across the entire genome. You can get non-disjunction at individual chromosomes. So it's either whole genome or single chromosomes. And there are cases of both of these things occurring. These gametes that then fuse with sperm, which are indicated by our tiny little capsules there, these will not be viable. In the same situation, when you have twice as many chromosome strands as expected in meiosis 2, your gametes are again not conforming to the regular amount of chromosomes and will again form triploid zygotes. Because after these two sets of chromosomes fuse with another set from dad, you will have material that is 3N rather than 2N. And so in a situation where non-disjunction occurs in meiosis 1, every single gamete, and therefore every single zygote possible, will not have a canonical or set number of chromosomes. If the non-disjunction event occurs in meiosis 2, you will have two zygotes that form up just fine, and two zygotes that form up with the incorrect number of chromosomes, one being triploid and one being haploid. So it really does matter when that non-disjunction is occurring. Now this phenomenon of triploidy, or if you have too little, aneuploidy, is really, really bad for a vast majority of human chromosomes. In fact, there are only 3 chromosomes that, if you happen to have one more or one too few copies, do you survive. In all other cases, these zygotes would be aborted. They would not be carried to full term. And in the 3 cases where that triploidy or aneuploidy is tolerated, you do get developmental defects. The best recognized of these is trisomy 21, which might be better known to you as Down syndrome. Now, as with the other trisomies, where there are extra chromosome counts, you do notice developmental defects and a shorter lifespan. But folks with trisomy 21 can live long and productive lives. In fact, we recently had our first individual with trisomy 21 complete an Ironman. That means that this individual ran a marathon, biked over 100 miles, and swam, I think it's a 5K, so it's 3.1 miles, all in one swoop. That's quite impressive for any human being, and doubly so for an individual with some developmental issues like those with trisomy 21. However, this chromosomal variation that applies to massive problems in autosomes is not the same in sex chromosomes. So your sex chromosome, aneuploidy, is better tolerated. And this has to do with the fact that we have genders determined by different chromosome counts. So, in humans, XX is female, and XY is biologically male. So how are males okay, since they have one chromosome and females have two X chromosomes? It's a dosage problem after all, right? If you have too many of a chromosome, you're making too much of a gene, and that can screw up developmental processes. So how come all females are not messed up? Well, it has to do with the fact that we are able to selectively methylate or quiet down an individual chromosome at a time. This method, called bar body silencing, named for the fact that the bar body is the chromosome that is formed when you methylate an X. It looks like a big purple dot when looking at it under a microscope. Basically turns off all the genes associated with that particular X chromosome. Now this results in some pretty cool phenotypes. For example, calico cats have their coloration pattern due to different pigments derived from either one X or the other, either white colored or color patterned with black or orange dots. This bar body silencing is something that's common across many different animals because animals are quite non-tolerant of ploidy variation. And it also allows animals to tolerate aneuploidy in terms of overall sex chromosome count. For example, an individual that has a single X chromosome and no second chromosome would still develop as a female. An individual with two X chromosomes and a Y chromosome would develop as a male because they have that testosterone gene. You can also get multiple different types of chromosomal variations with 3 X's and a Y or something of that sort. And there are different named diseases associated with these things, but I just want you to know that aneuploidy, at least in animals, is tolerated a little bit better than it is for the sex chromosomes than in the autosomes. Next up, we'll talk about some chromosomal issues. Rather than non-disjunction occurring and messing up entire chromosome counts, in the process of copying or in the process of recombination, you can get problems in the restitching of that chromosome back together. These chromosomal issues are oftentimes referred to as rearrangements. And there are 4 different rearrangements I want you all to be aware of. Deletions, duplications, inversions, and translocations. So, one at a time. If this is my chromosome, you will note that there are genes in a very particular order on this chromosome. From the black gene, to the purple gene, to the red gene, 2 on the other side of the centromere, pink to green to orange to brown. Whew. In the situation of a chromosomal deletion, you will see that a portion of the chromosome disappears. It broke off, it didn't convert, did not have that gene conversion opportunity, and therefore it's permanently gone. You, in this case, would encounter aneuploidy for this particular region. So deletions can result in localized aneuploidy. Duplications are instead where a region of the chromosome, as you might expect, rather than going away, it might double. So for example, here you would have gotten the black and purple region to duplicate on the end of this chromosome. And in this case, rather than causing missing genes, you instead encounter trisomy locally. So duplications cause you to double the amount of genetic information at that very particular region. So local trisomy versus local aneuploidy. Chromosomal inversions act a little bit differently. And actually, I need to correct myself real quick before we go on to chromosomal inversions. Aneuploidy is our term used for either one too many or too few chromosomes. Trisomy refers to when we have 3 chromosomes rather than a regular 2. Monosomy is our term for a single chromosome or a single gene where we're expecting 2. So monosomy and trisomy are opposite each other, and aneuploidy is the generic term referred to both of these. So deletions and duplications both result in aneuploidy, just different types, monosomy and trisomy. And inversions do not affect or do not result in aneuploidy at either the chromosome or the entire chromosome or the gene level. Inversions are instead where this chromosome will have a section of it. Let's say that top arm, rather than being arrayed black, purple, red, it will be red, purple in the middle, and then black, with the rest of the chromosome being just as expected. So the other markers down here will be the same. Now, why does this matter? It's still the same length DNA. There might be some small mutations as a result of the cutting and fixing, as we always expect. But what this means is that when you have this chromosome lining up with another chromosome that shares ancestry with it, you're still expecting the gene order to be black, purple, then red. Whether this was mom's or dad's chromosome, right? Because the genes and their locations are the same, but the alleles are different. In this case, the genes are different. The loci of those genes are different. This results in reduced rates of recombination. Because recombination and chiasmata want to form where sequences are homologous, where they're shared. In this case, by varying the genes that are there, you are now creating a small bubble. And so you might get this tiny little bubble that actually forms when the homologous chromosomes are lining up, preventing them from being able to recombine. So inversions lower recombination rates. In many cases, they can actually block recombination from occurring altogether. In biology, our Greek letter for recombination is rho, or the letter that looks like P. So in inversions, you're actually potentially creating blocks of genes that are going to have to be, or blocks of alleles that are going to have to be inherited together. So while it's probably beyond the scope of this class, this block inheritance is actually one of the things that we see consistently with different and repeated adaptation patterns. For example, you get these inversions or frequent inversions in fish when they're migrating from saltwater to freshwater, and those populations are then beginning to learn and adapt to live in freshwater. You'll oftentimes see large scales or numerous chromosomal inversions associated with adaptations to freshwater. Kind of crazy. I encourage you to read further about it if you're so interested, but that'll be it for this lecture. Now, a translocation will again not affect ploidy, does not result in aneuploidy, but it will again change recombination and ultimately change genome architecture. Because in a translocation, a whole piece of a chromosome, like the top half of the chromosome, might lop off. You just get deleted here, and you might get stitched on to another chromosome. So rather than having it be mom's one, dad's one, and mom's two, dad's two, you might get a whole piece of a dad chromosome that lops off and stitches onto another one of the chromosomes, could be dad's or mom's, but it attaches to the endpoint there. And in this case, you are again messing with the recombination rates and inheritance patterns and overall karyotype. But theoretically, you are not changing the genes or the gene dosage that is underlying our situation. So we have these 4 different chromosomal rearrangements. Those deletions that result in monosomy in a certain region, the duplications that result in trisomy, extra copies in a certain region, an inversion that lowers recombination rates locally, and a translocation that alters karyotype with an E at the end. Now, these chromosomal issues, whether they be aneuploidy or inversions or translocations, have different sensitivities based on which kingdom of organisms we're talking about. Because oddly enough, whereas deletions and duplications and non-disjunction events in animals are very lethal, quite often, in plants, they get a big old shrug. What's the big deal? Not a problem. In fact, this chromosomal flexibility, or this overall ability to deal with gene dosage, is something very unique and peculiar to plants, as far as we can tell for now. Still trying to understand what the heck's going on with the fungal kingdom. But we've spent most of our time understanding gene dosage in animals and plants, and we can see that plants do a much better job of coping with these massive variations or non-disjunctions. In fact, whole genome duplications are a feature, a regular occurrence in the plant phylogenetic tree. You see this happening tons of different times when you're looking at the plant phylogenetic tree. So I want you to know that there are variations based on the different animal or plant kingdom that we're looking at, but on a local level, all of what I stated about non-disjunction and chromosomal irregularities still apply. So at the end of this, we now have to ask ourselves, why? Why any recombination or reproduction via sexual reproduction in the first place? Well, fundamentally, this occurrence of recombination is useful to us because it allows us to combine adaptive traits. And by traits, I really mean alleles. And in this way, you have a much better or easier way of combining beneficial mutations that have been existing in parallel, meaning in different individuals, into a single individual to help them cope with future stresses. Compare this to, say, the parthogenic lizard. Those lizards that reproduce simply by doubling the genome of the female. Well, if there's a female who happens to be really good with underwater breathing as a lizard, and then there's another female who is really good at coping with dehydration stress, because of the fact that they never recombine, you can't combine both advantageous alleles into one individual. You can't combine both beneficial types of alleles at these two different genes into one individual. Asexual reproduction has to be highly mutation-driven. And we know that in general, most mutations are either neutral or nearly so, or the nearly neutral hypothesis. Most mutations don't really matter. There are more of them that have a negative impact than positive. So simply playing a numbers game, you make it much more difficult for a species to adapt to changing environments. We'll also end this section by doing a quick comparison of mitosis versus meiosis, just as a recap, because we're going to get into meiosis in humans and plants in a second here, so specifically looking at how this occurs in one group of animals, us. Mitosis has products that at the end are identical. You want cells to be completely the same. You want these cells to be even-sized. You want them to have the same genetic material. You want them to have the same organelles. In meiosis, all of your products are genetically distinct or genetically unique. And frequently, those products are of uneven size. And nowhere is that size more uneven than what we see in human meioses. So let's talk about this now. A little bit of human biology. So depending on your gender, you can perform meiosis in one of two different ways. We'll start with spermatogenesis, only because it is the less interesting of the two, quicker to get through. You begin with a stem cell for your sperm, these being your spermatogonia. And spermatogonia, are plural. Spermatogonium is the singular. And I also want to draw attention to the fact that if you ever have trouble with my handwriting on this, as you justifiably should, I apologize for my terrible handwriting, but you do have vocabulary sheets with all of these words typed out, so you don't have to question yourself on the spelling of them. So your spermatogonia can perform mitosis to replicate as they need to throughout your lifespan, and then they will eventually go through the process of meiosis. Now, the spermatogonia stem cell will at some point mature into a primary spermatocyte. And it is this primary spermatocyte that is a diploid cell that then copies itself to B4N to go through meiosis 1 and create a secondary spermatocyte. And if we're being technical here, these spermatocytes are technically a single end, but you know that there are two copies of them. So the secondary spermatocyte is a pseudodiploid, just of the same material. And then you have splitting again, where the secondary spermatocyte will mature into the sperm, where it is truly a genetically unique haploid. However, spermatogenesis generates 4 sperm, all of even sizes, of equal genetic contributions, of equal genetic makeup. This is most certainly not the case in egg formation or oogenesis. Just like in spermatogenesis, you always begin with the stem cell for this. The stem cell for eggs is the oogonia. The oogonia is what you have at birth. Now, these oogonia, or oogonium for singular, you start with something like 7 million of them. But at the time when you are born as a female, you will be down to something like 1 to 2 million oogonia, stem cells for eggs. So there's a lot of them, right? More than you'll use in your lifetime, more than you could possibly use in your lifetime. The oogonia will then, of course, just like in spermatogenesis, begin the process of meiosis. The first outcome here is our primary oocyte. Our primary oocyte will shoot off a polar body. Because unlike the genetically and size even sperm, eggs do not end up with the same size. The primary oocyte will shoot off a polar body of just genetic material and keep all of the cytoplasm for itself. This then leads to a secondary oocyte. And this secondary oocyte will stop at metaphase 2 in order for the egg to kind of reach a stasis point. Because the timing of these two steps, the primary oocyte and the secondary oocyte, are quite unique. So your primary oocyte can remain your primary oocyte for up to 50 years. It will remain dictate for up to 50 years. That means it's frozen in time, essentially. It's stuck with that polar body not yet released, just chilling, ready to go in your ovaries for up to 50 years, up until it reaches its signal to be released from the ovaries. in the release from the ovaries, you then will advance to your secondary oocyte, so that polar body is released only upon ovulation. Otherwise, that primary oocyte is hanging out in its dictyate state. The primary polar body will completely disintegrate in mammals. Essentially kind of dissolves. It's a small little thing that just is basically a little chunk of DNA that gets ejected. Now, the secondary oocyte will remain as a secondary oocyte, stopped in metaphase 2 after the egg is released from the ovaries, and only in the case of fertilization. Does the secondary oocyte become an ootid? The ootid is what's occurring when you have the secondary polar body being ejected and the correct chromosome count now occurring within the egg. This is happening after a sperm has already made its way in. Minutes after fertilization, the ootid forms, and then the ootid matures into an ovum that now can fuse with the sperm's nucleus in order to form the zygote. So it's absolutely crazy to consider that you have, as a female, cells that are essentially frozen in a dictyate stage in prophase 1 for up to 50 years. They then complete prophase 1 and go through the rest of PMAT 1, stopping at metaphase 2 once the egg is released from the ovaries. Then you only complete as many meioses as you have fertilization events in your life as a female. Think about how that differs in comparison of how many meioses sperm complete. How many meioses will a male complete? It's a very big number. This information is summed up well in a table from Wikipedia, so I encourage you to look there as oogenesis. And I do know, this is why I tell you to go to spelling online, not with my spelling, because of course I misspelled the genesis part of both of these. It's genesis, sorry for that. But you can go to Wikipedia and read a fair bit more about these two different processes. Very interesting, and I do hope that you are able to keep track of our oogonium, primary oocyte, secondary oocyte, ootid, and ovum, as well as our spermatogonia, primary spermatocyte, secondary spermatocyte, and sperm. This process is neatly illustrated in your textbook, where it shows that humans perform a whole bunch of cell division and mitosis in their diploid state after the formation of the zygote, or the fused egg and sperm. It forms these giant bodies that I call you and I. And then we have meiosis occurring within these two very particular organs, the testes and the ovaries, to make our sperm and our eggs that are haploid in their chromosome count. Note that there is no mitosis down here. Your egg and sperm are singular cells that do not live very long on their own once outside of your body. And that's the animal way. So of course it's not the plant way. We'll end today's lecture by introducing a very confusing but very important process in plants called the alternation of generations. And actually, I'm going to redraw this here. No need to have that here. The alternation of generations still requires us to alternate between diploid and haploid. So we have half the circle as our diploid state and half the circle as our haploid state. Meaning that the body here is 2N and the body here is 1N. And you will perform mitosis as a diploid in plants. And here's the headache. You will perform mitosis as a haploid in plants. That's right. These cells that are the result of meiosis, which are going to be your haploid cells, can then go off on their own, find a new address, find a new home, gather resources, and perform mitosis. They are their own bodies. They are their own organisms. The equivalents of your egg and your sperm are able to live on their own outside of the plant body, outside of the parents. It's very, very strange. Meiosis and the resulting haploid that is generated via a whole bunch of mitosis is undone through fertilization. So you have this loop between meiosis and fertilization then going on to create your diploid zygote. But you have this alternation between a generation of a plant that is diploid and a generation of that same exact plant that looks totally different, that is instead a haploid. By alternating these generations, we see that the life cycle here is totally different. The diploid generation we call the sporophyte. The haploid generation we call the gametophyte. And this distribution of sporophyte and gametophyte are not constant across all plants. The ancestral way, early land plants, things like mosses, well, they're actually gametophyte dominant. That means that when you are laying in a bed of moss in a forest on a beautiful summer day, you are actually laying on the sperm and egg body of the moss. The sporophyte, the equivalent of a diploid, looks entirely different to us. Mosses are gametophyte dominant, but that does not mean that they do not also have a sporophyte. And we'll talk about that in Bio 2. Other organisms, like ferns, well, they're actually sporophyte dominant, but they still have an independent and photosynthetic gametophyte, which adds a whole nother layer to this. Is that actually, in mosses, the thing that does most of the eating and most of the body growing, most of the mitosis, is your gametophyte. In ferns, it's the sporophyte, but there's still a fair bit of resource gathering that's done by the gametophyte. And they look totally unlike a fern. They're about the size of a nickel, or the size of, the nail on your thumb. They don't have any fronds. They're kind of like a little piece of green paper with maybe some tendrils coming out the bottom to pretend to be little root kind of things. They look totally different and totally weird, but they are still part of that fern life cycle. Every single fern that reproduces sexually will have to go through a sporophyte and a gametophyte stage. Sporophyte will be dominant, much more of the time is spent as a sporophyte, but the gametophyte is still present. By the time we get to some of our higher plants, like gymnosperms, or our cone-bearing and our flowering-bearing plants, the angiosperms, you have a sporophyte that's so dominant that the gametophyte is now nutritionally dependent on the sporophyte. So gymnosperms and angiosperms, or cone-bearing and flower-bearing plants, are much more like this system where the egg and sperm are reliant on living within or near the original parent than what we have in mosses, where the egg and sperm go off and form their own bodies. So there's a lot of variation that we have between plants, between genders in humans, and then all kinds of different animals that we won't focus on in this class or any of the biology classes I teach. But I do hope that this gives you an appreciation of those variations, and I do hope that you keep track of these different chromosomal variations and abnormalities that can go wrong with meiosis. It is important to understand these principles when talking to a genetic counselor if you decide to have children. You will be modeling these things out, and it will give you the opportunity, an understanding of these things will give you the opportunity to make better decisions in your own family planning. From here, we'll take a look at our Punnett squares. See y'all next time.